Stanford University.
And with that, it's a great pleasure for me to introduce Dr. Doug Sawyer. I've known Doug for many, many years, and Doug told me that this is the first time he's here to visit Stanford, so we're very pleased that he's here. And so by way of background, Doug received his undergrad, a bachelor's degree at Cornell University. Afterwards, did his MD, PhD at Cornell, and then did his clinical fellowship training in cardiology at Brigham and Women's Hospital. Afterwards, he became assistant professor at Boston University, and he was there for between 97 to 2006, I believe, and around 2007, he left to Vanderbilt, and he was the chief of cardiology at Vanderbilt between 2008 to 2013. And then afterwards, around 2014, he became the chief of the cardiovascular services at the Maine Medical Center, where he's been since then. And more recently, as of last month, he was promoted to be chief of academic officer at the Maine Medical Center. Doug has received numerous awards, and just want to name a few. He's received the AHA Established Investigator Award. He's been elected to the American Society of Clinical Investigators, also the Association of University Cardiologists, and also the Master of Academy of Maine Medical Center Institute for Teaching Excellence, which is a recurrent theme in his CV. That is, he's really passionate about the next generation of trainees. He's been PI on T32s and also K-12 grants. He's published prolifically. He's published more than 150 papers, and he's pretty much widely known as the person who elucidated the cardiac neuregulin ERB signaling pathway. And that's a signaling pathway that's very important for cardiac development. It's also very important because of the increasing recognition that many of the cancer patients, when they get chemotherapy, it hits upon that pathway, and then the patients that develop cardio-oncology issues. And so I would say Doug has done the seminal work in this area and is widely recognized for this particular contribution. And he's also shown that this pathway is quite involved in cardiac development and also in diabetes and also other stress conditions that may have an adverse effect on this pathway. And for that, he's going to come and he's going to give a talk on the Mencius paradigm for cardiac regeneration, although I couldn't figure out last week when I was talking about this slide what Mencius means, but I assume it's Chinese sage, right?
We're going to find out.
We're going to find out. Okay, okay. Thank you, Doug, for coming. Yeah.
Thanks, Joe.
Yeah.
Hi, everybody. It's really an honor to be out here, and thank you, Joe, for the invitation. Is David, are you out there? Where's David? Oh, no, David Pake. Where is he? David, good to see you. I think I owe this invitation to David, who invited me down originally. So I really appreciate the chance to be here at Stanford and to share the story with you. I've never really told the story exactly this way, so I'm looking for feedback. I have a few disclosures. I don't know if we give out CME for this conference, Joe?
No CME.
All right, so, but anyway, there are some disclosures. And I do want to say a word or two about Maine. In my new role as Chief Academic Officer, I feel like I have to be an ambassador to the state and the programs we support. And I don't know that many people know about Maine. Is Amanda Chase in the room? Yeah, oh, Amanda, your mom just sent me a note saying that you may be here. So I'm going to point to Amanda for any follow-up conversation about Maine, because her mom works in the PICU at Maine Medical Center, Cali. So Maine Medical Center, pictured on the upper right, was built and opened to patients in 1876, and it has expanded. That's the original front door of it, and you can't see the rest of the hospital behind it. It's the largest hospital to serve the state of Maine and northern New England, with about 650 beds. And our research institute is a small niche facility about 10 minutes south of Maine Medical Center, and that's pictured on the bottom right. And we're part of a healthcare system that is built to serve the needs of Maine with the stated goal on the last slide to improve the lives of the community we serve. With Maine Medical Center being the large facility in Portland, but extending up the coast. And for those of you who orient to Maine from a Bar Harbor or Mount Desert Island or Jackson Labs point of view, it's right out here on this little island. That's Mount Desert Island. We're down here in Portland, and the medical center affiliates with these other hospitals in the system. We train medical students in conjunction or collaboration with Tufts. We have a Maine track program. So we actually now make doctors in Maine, not just, for those of you who watched Dr. Dreamy in Grey's Anatomy. He's an actor. He was made in Maine, but he was an actor that plays a doctor. We actually now make doctors in Maine in collaboration with Tufts. We also graduate students in conjunction with the University of Maine and Tufts Graduate School of Medical Sciences. And is Tom Katermas in the room? And Lucy Liao, pictured here, trained with Tom back at Vanderbilt. And she oversees our training programs. So we are an old state. This is a population distribution of the United States on the far right. So older folks are at the top, so it has a shape to it that is not represented by Maine, where we have a bolus of older folks giving us at least according to that census, the highest median age in the country compared to, for example, Utah. And that has become a theme for us medically to help address the needs of folks as we get older. We are funded now by the NIH through a clinical and translational research network grant in collaboration with the University of Vermont, the University of Southern Maine, and New Hampshire that's focused on serving the needs of rural populations because the large majority of our folks, in fact, we are not just the oldest population, we're actually the most rural population in the country. So we tend to focus on work that relates to that. So with that, I'm going to transition to this talk and try to explain what the Mencius paradigm is for rejuvenation, as I like to call it, and see how you think about that. So Mr. S, and I don't know, David, you and I were in clinic and we saw some patients together, and I think this might have been one of the patients we saw. A 63-year-old gentleman at the time I met him, transferred into Vanderbilt University for consideration of heart transplant. He had a prior heart attack at the age of 44, single vessel disease. He presented late and was left with a big scar on the front of his heart. He worked as an engineer down in Texas at the time. He moved to Oklahoma and then later Tennessee. And he was lost to follow-up. So he presented, I didn't put the year up there, but this is about now, so 44 minus 63 is 19 years later, to a hospital with heart failure symptoms. Heart failure being noted by shortness of breath, a weight gain, so fluid in his legs and abdomen, and an echocardiogram done at the emergency room by the cardiologist in that other facility showed his LV to be dilated, left ventricular end diastolic dimension normally being below 52 in our lab, and an LV ejection fraction that was low, normal being in the 55 to 65% range. So he had systolic heart failure, preserved right ventricular function, and heart failure presenting late after an MI. Not unusual, although most people who had a heart attack at the age of 44 would usually get some follow-up visits in along the way to help them stay along the straight and narrow. So we took care of him. We diuresed him, got rid of fluid weight, and got him down to a dry, what we thought by clinical exam was a dry weight. And you can see by BMI, he's overweight, obese. He tolerated an angiotensin converting enzyme inhibitor, spironolactone, and a beta blocker, the standard therapy for heart failure. The transplant team said, you got to stop smoking before we can think about you for transplant, and we'll optimize your meds and see how you do. And as an outpatient basis, he had his meds titrated and did remarkably well. At 6 months coming back, which is when we would have seen him if you were there with me that day, David, he was biking 4 miles a day. He had quit smoking. He also quit drinking. He was a social drinker, not a heavy alcohol consumer. He lost 20 kilograms of body mass on his own, with the body mass now in the ideal range. Maximum ventilatory oxygen capacity, which is a measure of someone's maximum oxygen consumption during a metabolic stress test, was now in the normal range at 26, well above where we think about someone needing transplant. And his heart function had improved. His EF was, ejection fraction was now 35%, up from 15, and his heart had shrunk. It was now 62 So remarkable. He presents 19 years out from after an MI, gets put on medical therapy, does some things right in his life, and has cardiac improvement. Not complete recovery, but he definitely improved. So, and this is a cartoon of what you might say is going on with him. So here's his heart attack, gave him heart damage. His body reacts with the renin-angiotensin system, sympathetic nervous system. He develops progressive systolic heart failure. He gets put on neurohormonal blockers, the standard of care for systolic heart failure. And then somehow his heart recovers. These drugs don't make the heart recover. Something has to happen inside of his heart to help it recover. And I think that is clinically what we all hope for, but we don't know how to predict. We don't know how to help patients modulate. And so how does that work? We call that process reverse remodeling. I'm going to give it a different name in a second. It'd be great to know how it works and how we can promote it. So it was in this context that a nurse that probably you met, David, named Sherry, was in clinic with me, and one year she gave me an Eastern meditation philosophy calendar, and it had a daily Eastern meditation philosophy. And one day, Mencius was the quote of the day. And it was this quote, and I'm not a Chinese scholar, so I took it at face value. Someone might have misquoted it on this calendar that they put on my desk. But a great man is he who does not lose his childlike heart. And Mencius was a Confucian who lived a long time ago. That's someone's depiction of Mencius. I think he had a second name too. If you don't know Mencius, you can Google it. It's a knowable fact what his other name was. And it occurred to me as I saw that, and over time, thought about more. I'd been studying this idea of Mencius's for my whole career. And the idea that came out of it was, I call it the Mencius paradigm. It's basically a hypothesis that factors that regulate cardiac development, the childlike heart, and physiologic growth may promote reverse remodeling or myocardial rejuvenation, making the heart more youthful. Mr. S had rejuvenation of his heart, if you will. Hard to know about regeneration without having more details, but certainly rejuvenation. He had a more youthful and a more lively step in his gait and was looking forward to further life. When I was a postdoctoral fellow, as Dr. Wu mentioned, we began studying a pathway involving cardiac development, and this is the original paper that led us to study this. This was published in 1995, and it was published simultaneously with two other papers. I forgot to put the reference up, showing that neuregulin 1, as well as ERB2 and ERB4 receptor tyrosine kinases are required for cardiac development. And the specific requirement that this picture points out, there are other requirements too, is ventricular trabeculation. So on the left, normal left ventricle, or this is, I guess, single ventricle, AV cushion, atria, and the ventricle is trabeculating by forming extra cells that will later compact to form the left ventricular and right ventricular free wall. And here, the ventricle of these hearts that lack a functional neuregulin 1 or ERB2 or ERB4 gene lack trabeculation. And it was in that context that we began to say, well, what happens in the adult heart? To just give you, by way of background, the neuregulin and ERB receptor family is really part of the EGF and ERB receptor family, EGF being the original growth factor described to interact with the EGF receptor, which in this nomenclature is ERB1. And neuregulin, distinct EGF-like ligand, usually transmembrane receptor, but also can be secreted, interacts with the receptor tyrosine kinases ERB3 and ERB4, which can homodimerize ERB4 or heterodimerize to activate intracellular signaling. In the developing heart, what those three papers basically showed was that the ventricular endocardial cells are neuregulin expressing cells, and the ventricular myocytes are neuregulin responsive cells, with the responses being tissue morphogenesis in the form of ventricular trabeculation. So that's the developmental paradigm that we set about thinking about whether that signaling system has any role in the adult heart. And at that time, in the lab of Ralph Kelly and Tom Smith at Brigham and Women's Hospital, we just started to look at a recombinant form of neuregulin known as GGF2, which I'll come back to, whether it activated receptor tyrosine kinases in a neonatal rat ventricular myocyte or an adult rat ventricular myocyte. And sure enough, the receptors were there. They responded to these ligands. And if you left for the weekend, so being a husband and father, I would set up experiments that worked around my social obligations, if you will. And we were trying to figure out what neuregulin did to these cells, and the experiment that helped one direction was leaving for the weekend to go take care of the kids and coming back the following week. And the cells had started to grow from the former intercalated discs and actually reform connections over a period of time. So that is one of the in vitro growth responses you can see with neuregulin. And in general, if you cartoon a bunch of work that I'm not going to go through, you can say that in the adult heart, the microvascular endothelial cells are at least one source of neuregulin, and ventricular myocytes are one cell that do respond to neuregulin, and the responses, you could lump them as perhaps tissue maintenance and repair, and we'll come back to that. But they include in vitro myocyte growth, survival, contractility, glucose uptake, mechanical coupling, and we'll come back to whether proliferation is a potential response. So to bring together a whole bunch of work that I'm not going to go through, I would like to suggest that neuregulin signaling in the postnatal heart is a local maintenance, repair, and plasticity system. It's activated by stress, and it helps respond to stress. It's part of a plasticity system, too. There's a group in Belgium that has shown nicely that the cardiac growth that takes place during physiologic growth of the female heart during pregnancy is mediated in part by this system. And there's investigators that have looked at whether the exercise-induced growth of the heart is in part activated through this system, and it appears to be. So it's a local plasticity as well as maintenance and repair. And as Joe alluded to, and I won't get into it, we've seen extra evidence of this through the oncology world's focus on the ERB2 oncogene, where targeted therapies such as the antibody trastuzumab can promote a cardiac dysfunction phenotype. And again, I won't get into that, because I want to get into the development of therapeutics around the idea. And all of this led to a lot of effort by us and others to think about whether we could just, instead of figuring out how to make the system better in someone's heart, like Mr. S, could we just give them back some extra recombinant human neuregulin and find something beneficial there? So to summarize a lot of work over a lot of years, and there's another group in China that's done this as well, and I'm just going to show our data, but we get a similar response. Whether you take rats after a heart attack, and LV fractional shortening is on the y-axis in each of these graphs except for this one. So after a heart attack, early after a heart attack, 1 month, there's improvement in heart function with neuregulin. If we wait till later when the heart gets progressively dysfunctional, we still see a response. If we take diabetic rats, and I'll come back to this if we have time, we had a hypothesis that perhaps diabetes was a state of neuregulin resistance, so we wondered whether we'd get the same result. We did, although it's delayed compared to a non-diabetic rat. Pigs after a heart attack as well show improved systolic function. This is actually, these are the untreated versus the treated pigs. Not as robust response, but a response nonetheless. One of the things about pigs, so who works with pigs here in the room? Anybody? Joe, how fast does the pig grow?
We use mostly mini swine.
Oh.
Yeah.
Yeah. So these are farm pigs.
Yeah, they grow a lot. Yeah.
So over the course of this 35-day experiment, these pigs grew about 30 kilograms. So this is not your average patient out there, and it's bred to grow skeletal muscle. So we didn't see as robust an effect, but it's complicated by the fact that these are changing animals. And I'm not going to walk through this because I'll come back to it, but all of this work led to the idea that maybe we should do this in people. And I'll walk through a study that Dan Lenihan ran at Vanderbilt. We worked with a company called Accorda that took the GGF2 form of neuregulin and made it into a GMP-compliant product that the FDA approved for us to do this study with. And they named it semaglutide to complicate things. So now neuregulin is, in Accorda's hands, is now known as semaglutide. And we gave them a 30-minute infusion of an escalating dose protocol. We designed the study protocol around the animal work we had done. We chose 7 doses, escalating dose, 6 people in each cohort. 4 people got randomized to study drug, 2 to placebo, blinded to all. And it was an escalating dose to an endpoint of toxicity or finishing the protocol. And in the 7th cohort, we reached a toxicity of a transient elevation of liver transaminases and bilirubin, which ended the protocol, so hence we didn't get to the full complement of subjects. And it was showed safety up until that last dose, and in it we looked for efficacy signal, really to power an efficacy study. And what was different about this study, other than all of our animal work to date, was that we were only giving a single dose for safety purposes, and in all the animal work we had used repeated dosing. So the rat studies I showed you, they got two doses per week. The pigs got 2 doses per week. So this was different. We didn't expect a signal, but yet what we see is, I'm going to walk through this, is that folks in the high dose cohorts, when you combine them, showed an improved change in LV ejection fraction that occurred within a week to 2 weeks of infusion and appeared to be sustained. The numbers are small. These were all blinded. They were read by people blinded to the subject allocation. The placebo group is here in gray, and the low dose cohort really just has noise. So it would appear that at least in people, a single dose of neuregulin is associated with improved systolic function. I can see some scratching of the heads, and we scratched our heads about this too. When I go back to this animal work, in each animal we saw a different signal for why it mattered. So for example, in the rat study, we did microarray analysis and proteomics looking for what's the signal about, and the data pointed towards perhaps we're inhibiting oxidative stress, raising the levels of some of the antioxidant enzymes that have been implicated in heart failure. In the pig study, we did microarray analysis and had to annotate the pig genome to analyze it fully, and saw signals for an antifibrotic effect in pigs, as well as an activation of developmental programs. In the human study, we didn't have that opportunity. We didn't have the chance to see what happened to the hearts of those people except by ejection fraction. So we're left puzzled by this. What's interesting is it's not that different from a study done by the group in East Asia where they gave a different form of neuregulin over a course of 11 days at the beginning of a protocol, and they see, so 11-day infusion, daily infusion, 11 days of it. And then they watched, and in those subjects that were treated with neuregulin, they also saw a sustained improvement in LV ejection fraction. So a single dose with a different compound. So their neuregulin is cartooned up here. It's a small, this is the full cartoon of neuregulin with all the different splice variants that are shown in gray are expressed in the heart. And EGF beta would be the minimally active form of neuregulin that's used by the company's ensign. And the GGF2 variant that we were using is a larger molecule that has an Ig and a Kringle domain that appear to have some matrix interacting properties that perhaps gave it a longer half-life. And so one argument you could make is that both studies show a single dose in humans gives a sustained improvement in cardiac function. But we really don't know why. We had ideas I showed you in the beginning about myocytes, which is how we really got into it. When we studied the pigs, we started thinking about, well, perhaps it's fibroblasts are helping to drive this because of their effects of neuregulin on the the extracellular matrix that we saw. And if you look up that paper, you can see more details. And then in the human study, I won't get into the details of it, but we actually collected peripheral monocytes from the patients in that study that Dan ran, and we looked at an inflammatory response because of some data we had that showed monocytes express ERB receptors. And in a paper we published in AJP that seems to be true, that people treated with GGF2 had a suppression of TNF-alpha, and in vitro you can show an anti-inflammatory effect of neuregulin on monocytes. So maybe there's something else, maybe there's another mechanism. When we first started the work, I was sure that this whole idea was going to fail because we were giving the drug or the compound systemically, and the big concern of systemic growth factors is that we might promote cancer growth or other tissue growth that would be bad for patients. And in fact, in the GGF2 protocol, the FDA required us to use a cohort of patients that had heart failure so that we would shift the risk-benefit ratio, i.e., normal human subjects shouldn't be given GGF2 because they don't have any likely or potential benefit of it, whereas a heart failure patient, based upon the animal data, might derive some benefit from it. So I'm now convinced that maybe it did work because we had a systemic effect, i.e. we did something else that helped reset the human biology in a way that might have led to that sustained improvement if it's in fact reproducible. So now there is a group out there that, or maybe more than one group, that has shown proliferation of myocytes in response to neuregulin. And so you could say, well, could we have done some myocardial regeneration? This is Bernard Kuhn's work. This was when he was at Children's Hospital, although I think he's in Pittsburgh now. And to just focus over here. So in mice, they showed an improvement in cardiac function, and they showed a reduction in scar area. And so perhaps there's another mechanism out there. In rats, we did not see evidence for that effect. In this study I showed you earlier, we also did PET imaging serially in these rats. So you can see that this is a short axis view of the myocardium that has FDG, radiotracer uptake. And in the infarcted animal, you see there's a big scar along one side of the heart. And at least by this measure of myocardial viability, we did not see any change in that scar, either in terms of its ability to uptake FDG or in total myocardial FDG uptake. So at least by that method, you could debate that, whether it's a good method. Regeneration wasn't at work in this experiment. When you look at all the different effects that neuregulin has, you can cartoon it here. This was a paper we wrote a while back, but we've talked about physiologic adaptation and hypertrophy cardiac myocyte. There is evidence that angiogenesis is also regulated by neuregulin, and that comes out of work by Headley, who was up at Yale at the time. There's also skeletal muscle effects of neuregulin. And in our pig work, we're pretty sure it regulates fibrosis and perhaps monocyte inflammation as well. But we really don't know of all these things or others what happened in those human hearts. I'll come back to this cartoon and say, well, going back to Mr. S that I told you about earlier, is it possible that neuregulin signaling was part of his recovery, his own endogenous neuregulin? And we can tell you that he did take up an exercise habit that could have changed his dynamic neuregulin. He also changed his eating habits to lose weight. And this signaling system is influenced by diet and exercise in a way that might say, yeah, that's a reasonable hypothesis, but we don't have any way of knowing that right now. So here we are, 1995 to 2015. It's taken us 20 years to put neuregulin back into people after somebody showed that it was critical for cardiac development. So we're not moving that fast. We might be able to do better. I don't know. We still don't know if it's a good therapy. We can talk about what the future might hold there. And if it is a therapy, we really don't know how it works. So I want to spend a few minutes talking about how we might change the dynamics a little bit and how we think we might change the dynamics in order to accelerate that paradigm. One thing we tried to do early on in the neuregulin program, but we weren't able to do, was what we might call a phase 0 study, examining how biological responses are affected by neuregulin. Is anybody familiar with a phase 0 study in oncology and what that looks like? Okay, so phase 1, is that safety trial we talked about. Phase 0 has been dubbed this idea that you take a potential drug that you've shown is effective in your cells or your mice or other animals models, and you ask whether it has the same biologic effect in people. So if I have a tumor that's going to get removed by a surgeon next week, and you're going to take that tumor out as reductive therapy, you've already proven I have a tumor, then you could sign me up for a study, and I might even do it, that says, well, you have a new idea for a new drug, and you know it's safe for me to take. Give it to me now, and when you take my tumor out, see if it did to the tumor what you saw it did to the tumor in mice. And that would be a phase 0 trial, looking for signal of biology or therapeutic signal. And we really don't have a strategy in cardiovascular science to do phase 0 trials. If we did, we might have been able to give people neuregulin and then look at their hearts early on and figure out what dose would be the right dose to affect the heart and what would be the biology that we'd be trying to affect. So you could imagine there might be some scenarios where you can do a phase 0 trial. We proposed to the company the first trial we would want to do is to give a patient neuregulin a week or whatever number of days, we could debate that, prior to a ventricular assist device implant. And at the time of ventricular assist device implant, anybody work with tissue from ventricular assist device patients, we take out an apical core, and so you'd have an access to a sample that's part of the routine clinical work and would not be any extra risk to the patient, but for the exposure to neuregulin. There was too much concern over a couple of issues with that idea. So we also thought about a serial biopsy study where we put people through a biopsy of their heart, which we do clinically for diagnostic purposes, and then come back to them with a second biopsy after giving them an exposure to neuregulin. And we went through an IRB-related review of that, and that didn't really float. So we really didn't get to do what we ever wanted to do with neuregulin phase 0, but we thought we ought to go about creating a pathway for phase 0 studies in cardiovascular disease. So we talked about the ventricular assist device as one opportunity. It's discarded tissue. One of the limitations is it's advanced heart failure patients. These are pretty far advanced, usually listed for, we're about to be listed for transplant unless they're not transplant eligible. There is another opportunity, and that is any patient undergoing open heart surgery under the right skilled hands can have an epicardial biopsy that is minimal risk according to some of our colleagues up at University of Vermont. And it has the pros of we could take people without advanced heart failure or heart failure going through mitral valve disease or ischemic heart disease surgery. The con is it's a biopsy that isn't part of the planned clinical care, and so it does add risk to that patient's care. In any case, we talked to surgeons about this and worked through some of that discussion to develop that protocol up at Maine Medical Center, and used it to, at first, proof from principle we could do it safely, and in second case, look at whether there are cells in the heart that we weren't paying attention to that might be related to some of the data I've shown you. So here's a graph of, well, there's a lot of graphs on this page, so let me limit you to this up here. So this is the weight of the biopsies of a number of patients undergoing biopsy of an epicardial sample about the size of a grain of rice that are IRB approved with careful watching. And the biopsies would come down to the lab. They weighed between 10 and 35 milligrams. We were able to isolate viable cells from them and do flow cytometry separation of them to look for, in particular, we were looking for these CD105 positive, CD45 negative, CD31 negative. So CD105 being endoglin, being a marker in some people's view of progenitor cells that are not hematopoietic and are not endothelial cells. And so that was the initial protocol. We went on, Sergei Razov went on to demonstrate that from these biopsies we could isolate, he ended up dubbing them highly proliferative cells from the epicardium that express ERB receptors. They vary in their expression of ERB receptors that are stable over many passages, and this is published. Over here, unpublished, these cells have an excess abundance of some transcription factors that some would say make them more stem-like or progenitor in nature. And Sergei went on to show that if you took the cells that were high in ERB expression versus low in ERB expression, they had differences in their ability to do angiogenesis or endothelial-like assays. This is a capillary-like tube-length formation assay, and over here is a dextrane fluorescence diffusion across a monolayer assay. And the high ERB2 cells are more endothelial progenitor-like than the low ERB2 cells. And with help from Rong Li Liao, we are now taking those cells back and asking whether they're different when we put back them into a nude or a NOD skid mouse that lacks an immune system that can react to them. So thank you, Rong Li, for your help with that. These are high ERB2 cells injected back into a mouse heart 7 days after MI versus saline. And like other cells, they are associated with improved fraction shortening. And the goal of the lab right now is to figure out whether the high versus low ERB2 matters. And I went on that tangent for Rong Li's sake because I had to show you that we've been responsive to your help. So come back to this diagram, and we got to add some other things to it. So we had cardiomyocytes, we had CD14 monocytes. I took the fibroblasts off just simply because I needed space to add endothelial cells and then perhaps progenitor cells that might be involved in the response to neuregulin. And without being able to do that phase 0 trial, I don't know which one might be playing a role in the results we saw clinically. So come back to Mencius. And what did we learn? Well, one thing you could say we learned is that it was reasonable to pursue the idea that a developmental signaling system could be helpful in the failing heart. And so something critical for cardiac development is worth considering as a modifier of adult heart function. And I'm going to dub that rejuvenation, which I know is an undisciplined, perhaps, use of the word rejuvenation. And I wish I could come back for your Thursday session about telomere length so I could be more sophisticated. So if you go back and say, well, what else could be like neuregulin in that regard, and what might we spend some time and focus on? And this comes from a review that's a few years old now, and so this is not a complete list. But among these things, notch, ephren, BMP, and specifically 9 and 10, SEMA6D, ATOM, I forget what the TS stands for in ATOM TS. I think it's, anybody know? No. Cypher's ASP and retinoids. We spent some time ourselves looking at retinoids, but we think this paradigm deserves a more careful look at these and other systems that are involved in this phase of cardiac growth and development. And so with those cells in culture, that's one way to do it. We've got human cells now. We're taking that approach with the retinoids and BMP right now, BMP in collaboration with Calvary, retinoids in collaboration with Marianne Asen-Beatrice, who's a retinoid biologist interested in regeneration. And perhaps with that paradigm, we might accelerate our path. The other thing to think about is whether the phase 0 clinical trial is worth pursuing with more rigor. So in order to get to the point where we got the biopsies, we had to get engaged surgeons in the room. So Mike Robich is shown here. We recruited him from Cleveland Clinic. He worked with Frank Selke as a postdoc fellow during his surgical training. We had to be in a culture that had willing patients, and luckily we live in a part of the world that really appreciates the chance to be involved in clinical research, to the point where I went in to do weekend rounds last month, and a patient who had enrolled in one of our studies 6 months ago, who I had never met, I had met part of our team and our clinical team, but I just never coincidentally had met him, looks at my name and says, oh, yeah, how are my cells doing? So he cared about what was going on with what happened. He remembered enough to ask the question. We needed a busy surgical program. Our program, I think, reasonably busy. We do about 1,200 open hearts a year. And we needed a research infrastructure. And as I mentioned, MCRI is our research infrastructure. And then we need some hypotheses worth pursuing with passion. Now, with our neuregulin, we had passion, but we were working with an industry sponsor and the FDA that didn't let us go down the path of a phase 0 trial, and that's okay. That was ultimately their decision to make. But there are other ideas out there that we don't need an industry sponsor for that might relate to rejuvenation. And perhaps we can apply this phase 0 concept to them and learn more about the human response to different things. So I'll give you 2 examples. So how many of you have heard of the Mediterranean diet? Was your lunch compliant with it, just out of curiosity? No, okay. So Mediterranean diet is meant to, is thought and is proven according to the PREDIMED study to reduce risk of adverse events, cardiac MI, death, stroke in patients that have diabetes or significant risk for heart disease. This is that the event graph from that trial that has been published a number of ways because they had to reanalyze it recently. But basically it showed that if you gave people instruction over Mediterranean diet and extra virgin olive oil or a nut supplement, that they had a lower event rate. Now, the mechanism by which this affects the human heart is not clear. It's not, you can suppose and you can hypothesize, but it's never really been looked at carefully. So we wondered, and of course you could debate this, but these people might be rejuvenated by the Mediterranean diet. Okay, so if rejuvenation is your goal, and this might be a non-Mencius strategy for rejuvenation. So we had been interested in how diet affects signaling of neuregulin for quite a while, having studied it for a long time and recognizing that insulin signaling is a lot like neuregulin signaling. So why isn't insulin resistance just a state of neuregulin resistance? It's just insulin signaling through its pathway to activate glucose uptake, and neuregulin activates glucose uptake via a very similar pathway. And in fact, Tom Miller, who's now a congenital cardiologist in Utah, showed some years ago when I was with Ron Lee at BU that palmitate can disrupt cell death and oleate can rescue it, oleate being the active fatty acid in olive oil. And this was associated with disrupted neuregulin signaling. So neuregulin, which normally would rescue cells, actually became more pro-apoptotic in the setting of palmitate, but the signaling was restored by olive oil or oleate. So that was exciting to us, and we never quite knew what to do with it. Ilka Pintz, who's up at Maine Medical Center Research Institute, had been studying palmitate's effect on cardiac function for some time. And she showed that a high palmitate diet, which is one of the ways in which you disrupt insulin signaling to create insulin resistance, is associated with a reduction in rate pressure product induced by calcium in an isolated Langendorf preparation. So we put our heads together and said, well, why not think about whether a phase 0 trial might make sense? We got a hold of atrial appendage tissue from that protocol I've been talking about, and she did lipidomic analysis and showed that diabetics, and this is a principal component analysis of lipidomics from these patients, left atrial or right atrial appendages, non-diabetics separated out from diabetics, and one of the primary components were a reduction in outer leaflet lipid components that might be associated with disruptive calvuli, which is one of the signaling mechanisms by which these receptors stay coupled to their signaling system. So it led to this idea that perhaps Mediterranean diet, modified Mediterranean diet in our case, because Mediterranean diet allowed you to drink alcohol, and as I will go into, we didn't include that in our modification, might restore ligand signaling and lead to improvement in cardiac function. So to test this idea, we proposed and are now open for enrollment a phase 0 trial where we take patients who are admitted to Maine Medical Center but can't go to cardiac surgery for a week because they have to go through a washout of an antiplatelet agent, which we won't get into, but we can talk about. So they're stuck with us, and they have to eat our food. And there's a subset of our menu that is compliant with the Mediterranean diet, and our nutrition services people were willing to add some things to it, an extra fish component, extra nuts. And we now have a randomized trial supported by nutrition services looking at whether when they go to surgery and we take out a biopsy of the heart and look at endothelial function as well as lipid components, that diet had any biological effect in the heart. Really, A, looking for biological response to diet, but B, trying to develop a proof of concept around a phase 0 trial looking at biological signals. That's ongoing. Hope to say what we learn in a bit. A second trial that I'll close with is Mike Robich's trial. He had studied resveratrol, what's thought to be the active component of red wine in some people's view of the world of red wine being good for you. Resveratrol in animal models restores a lot of things, including endothelial function, but has never really been looked at in how it affects the human heart. So Mike is now halfway through recruitment into a phase 0 study of resveratrol versus placebo in diabetic patients undergoing open heart surgery funded by the AHA. And we hope to learn whether, at least at that dose, resveratrol had any activity in the human heart. I don't know what the future is for resveratrol, but we think this will be the first phase 0 trial in cardiovascular disease, setting up a paradigm for perhaps finding a more efficient way to go through drug development. And if you think about
Stanford University.